Very good evening to all the NEET aspirants. Today we are going to unlock all the diagrams of NCERT 11th standard. Let's decode all the diagrams quickly and let's score higher in the exam. Okay, first from the chapter biological classification. Biological classification. So in this, if you look into different shapes of the bacteria like cocci, which is spherical, bacilli, which is rod shaped. Most of the pathogenic bacteria are of bacilli type. Most of the pathogenic bacteria are of what type ma? Rod shaped, those are bacillus type. Next, spirilla. Spirilla are of spiral shaped. They are spiral shaped bacteria. And vibrio, these are comma shaped. Vibrio are comma shaped. If you look into the singular terms of those different shapes of bacteria, this is coccus, bacillus, spirillum and vibrium. And then if you look into Nana, cyanobacteria, which is eubacteria, very important diagram where you have to fix it in your mind, which is also called as blue-green algae. So in this, if you clearly look into beta, cyanobacteria has two types of cells. One is vegetative cell, one is, just a second, vegetative cells. Second is heterocyst. Second is what type, Nana? Heterocyst. Okay, so this vegetative cells will have both photosystem 1 and photosystem 2, and they have chloroplasts which are embedded at the surface of the membrane as colored pigments, where you will call it as chromatophores. And if you look into the cytoplasm in the vegetative cells, you can observe central colorless centroplasm, and peripheral colored chromoplasm, And in vegetative cell, oxygenic photosynthesis, it conducts oxygenic photosynthesis, and electron donor is water. Water is the electron donor. And second type of cell, if you clearly look into heterocyst, these are thick and tough walled. Which are impervious for gaseous exchange. These are impervious for gaseous exchange. Okay, because these cells will conduct only one activity, that is nitrogen fixation. Okay, and nitrogen fixation is done by the enzyme nitrogenase. Nitrogen fixation is done by the enzyme nitrogenase. This nitrogenase will be inactive in the presence of oxygen. That is why the walls of heterocyst are very thick and they are impervious for gaseous exchange. So these are the two types of cells in cyanobacteria, and this cyanobacteria are covered by gelatinous sheath. It is covered by gelatinous sheath. Okay, and it is a prokaryotic cell. Best examples of cyanobacteria are Nostoc, Anabaena, Olocera. All these comes under cyanobacteria only. Okay, and next if you see beta, binary fission. Okay, that is the asymmetric division of a bacterial cell, where you will also call it as amitosis, where mitotic division or division of two cells happen without spindle apparatus or spindle fiber formation. Okay, this usually takes place in bacterial cell, which is a normal cell division, where you will call it as amitosis. Okay, and next if you look into Nana, these are protistans, which are all unicellular eukaryotes. They are all unicellular eukaryotes. Okay, all these unicellular eukaryotes, you have to quickly identify all those diagrams beta. First, this is dinoflagellates. Dinoflagellates. Okay, these are photosynthetic protists. Let us quickly remember their features as well. They have stiff cellulosic cell walls, one longitudinal flagella and one transverse flagella, both together will form whirling whip movement. And those dinoflagellates which show bioluminescence like noctiluca, and those dinoflagellates which form red tides in the Red Sea like goniolax. And then here if you see the clear diagram, that is fungi like protists, best example is slime molds. Fungi like protists, those are slime molds. Okay, these are wall-less, but the spores that are produced of the slime molds have wall, right? And then it is euglena, this is paramecium. Both of them come under protozoans. They both are what, Nana? Protozoans. Okay, sorry, euglena is a photosynthetic protist which has mixotrophic nutrition. They have mixotrophic nutrition. Okay, whereas paramecium is a protozoan which are animal-like protists. And this is a plant-like protist only, but then there is no cellulosic or stiff cellulosic cell walls here. The cell wall is absent, but euglena possess a protein-rich layer called as pellicle. Okay, euglena will possess a protein-rich layer called as pellicle. Okay, protein-rich layer called as pellicle. This pellicle will show different movements of the euglena where they can change their shape. That is why it is also called as, that movements are called as metabole, which are facilitated by the protein rich layer, that is pellicle. And from this paraflagellar body, one short flagella and one long flagella will form. Okay, one short flagella and one long flagella, which is where you will call it as trichonematic flagella. And you can see one contractile vacuole in the euglena. This contractile vacuole is helpful in osmoregulation. This is helpful in osmoregulation. Clear beta? Done with the euglena. Now when you look into paramecium, when we are learning about cell theory also we have learned, as it is binucleated, cell theory is not applicable to paramecium. And here in paramecium you can see one large macronucleus and one small micronucleus, and the contractile vacuoles which are present in the paramecium are also helpful in osmoregulation, and these paramecium comes under ciliated protozoans. They come under ciliated protozoans. Okay, where the body is completely occupied with or surrounded with thousands of cilia. Okay, and then if you look into beta, here if you see, these are different pictures of fungi. You have to identify it. First one, what is it? Rhizopus. This is Rhizopus. Okay, which is a bread mold that comes under Phycomycetes, right? And this is Aspergillus. Aspergillus, which comes under what class, Nana? Ascomycetes. And here this is Agaricus. Okay, this is Agaricus, which comes under the advanced fungi, most advanced fungi, that is Basidiomycetes, that is Agaricus. And then if you see, lastly, viruses. Okay, two different types of viruses. This is tobacco mosaic virus, and here it is a bacteriophage. Okay, this is a bacteriophage. Okay, tobacco mosaic virus, which is discovered by DJ Ivanovsky, where you have a strong protein coat that is capsid. This protein coat has many protein subunits, where you will call it as capsomeres. Many protein subunits, you will call it as capsomeres. Okay, and As it is tobacco mosaic virus which is affecting a plant, you can remember it that way. This is RNA, that is single stranded RNA. Okay, it is a single stranded RNA, and you can observe a protein code, that is capsid, where you have many protein subunits. Those are capsomeres. If you look into the nucleotides that are present in RNA, you can observe 6400 nucleotides. Here you can see 2130 capsomeres approximately. If you take the ratio of them, it is nucleotide ratio versus capsomere ratio. If you see, that is 3:1. Okay, and then here if you see Nana, bacteriophage, you can clearly observe head, collar and tail fibers. Okay, head, collar, that is sheath, and then tail fibers. At the end of tail fibers, you can clearly observe there is an enzyme which is called as lysozyme. The enzyme is lysozyme. So here at the head, the genetic material is present which is delivered through the collar into the bacteria, and this tail fibers will attach to the bacteria which will lyse through the enzyme lysozyme, lyse the cell wall of bacteria through lysozyme. Coming to the plant kingdom, beta, in plant kingdom if you see, we have different types of algae. Okay, plant kingdom if you clearly observe, we have algae, different types of algae. First is chlorophyceae, phaeophyceae, second is phaeophyceae, third is rhodophyceae. Chlorophycean members, the very first chlorophycean members, the diagrams which are given in our NCERT are colonial volvox and filamentous algae, filamentous green algae, that is Eulothrix. Okay, this is colonial volvox and this is filamentous green algae, that is Eulothrix. So here if you see, these are the daughter colonies which are present in the main colony of volvox, and these are the filamentous Eulothrix. And second if you see, brown algae. Okay, brown algae which comes under phaeophyceae. Three you can see under brown algae, that is, first this is laminaria, and here you can see fucus. How you can identify fucus, beta? By air bladders where you can see in the front. Okay, these air bladders you can also call it as pneumatocytes. And here if you see, third is dictyota. How you can identify the diagram of dictyota, Nana? By dichotomous branching, and only in laminaria you can see holdfast, stipe and frond. There is no stipe in fucus and there is no holdfast in dictyota. As such, you can memorize the brown algae diagrams. Next, when it comes to red algae, or rhodophyceae, one is porphyra and next branched form, that is polysiphonia, where you can find main axis and branches to that. Okay. Next, these are bryophytes, which are the amphibians of the plant kingdom, where here these are liverworts, that is Marchantia. This is Marchantia. Marchantia is unisexual or bisexual. This is bisexual or monoecious. Bisexual or monoecious. Sorry, this is unisexual, right? Marchantia, which is unisexual or dioecious. Okay, wherein you can clearly observe the Gimma cups which modulate vegetative propagation, and you can clearly see this finger like projections out. You can observe it as archegoniophore. If they are little inward, you can identify it as antheridiophore, and as the dominant plant body is haploid, rhizoids, Gimma cup, thallus and antheridiophore, archegoniophore, everything is haploid in liverworts, that is Marchantia. Coming to here, you can clearly see this is the diagram of funaria. And what is this, Nana? Sphagnum, right? Which is also called as peat moss, bog moss. Many at times the questions are asked based on the commercial importance of sphagnum also. What are the common names, like other names for sphagnum? Peat moss, bog moss or cotton moss. Funaria, if you see, you can observe multicellular rhizoids and leaf axis which is haploid gametophyte, and from this diploid sporophyte will emerge out. Diploid sporophyte. Diploid sporophyte will include foot, seta and capsule. Foot, seta and capsule are diploid. Okay. Foot, seta and capsule are diploid. Capsule consists of spore mother cells. Okay. Capsule consists of spore mother cells. This will undergo reductional division, which is also called as meiosis, and they will release haploid microspores or haploid spores. Okay, this is the diagram of funaria. Many at times asked, ma, focus on this diagram 2 to 3 times. Next comes is pteridophytes, which are horsetails or hornworts. So these pteridophytes, if you see, pteridophytes you can observe. First is the diagram of Selaginella, which comes under which class of pteridophyte, Nana? Lycopsida. This is equisitum. This is equisitum, which comes under Spinopsida, and this is Salvinia. Salvinia, and this is Ferns. Okay. So Selaginella has small microsporophylls and macrosporophylls. The leaf-like structures called as microsporophylls or macrosporophylls because they have different types of spores. Both Selaginella and Salvinia, they will show heterosporous condition. Heterosporous condition. Okay, 2 different types of spores are formed, and these spores are present on over the leaf structures. That is why leaves are called as sporophylls. In equisitum, if you clearly see, Nana, spores are arranged into compact structure called as cones or strobili. Okay, do remember that. And next, if you see, coming to gymnosperms. Gymnosperms, this is the picture of cycas, this is pinus, and here it is living fossil where you can find fan shaped leaves. Important. Okay, where you can find fan shaped leaves, those are ginkgo biloba. This is ginkgo biloba. Okay, and few characters with respect to cycas and pinus. Cycas root system is in association with BGA, that is cyanobacteria, where it forms coralloid roots. Pinus will form mycorrhizal roots where the roots are in association with fungi. Here stem is branched and here in cycas it is unbranched. Okay, do remember. And then if you see, when it comes to morphology of flowering plants, very important, where you can expect a diagram this time from the morphology of flowering plants chapter. So here the first basic diagram which shows the vegetative characters and floral characters clearly. Okay, vegetative characters include root, stem and leaf. Floral includes like flower, fruit. Fine. So, and this above, positively phototrophic ice shoot system, negatively phototrophic and positively geotrophic, that is root system. If you clearly see, ma, regions of the root, highly important. Okay, regions of root. Okay, first is root cap, which is at the apical region, apical or terminal region. Okay, small thimble cap like structure at the apical or terminal region of the root is called as root cap. Okay, root cap is rich in Golgi bodies. Proximal to root cap, you can find region of meristematic activity. Okay, proximal to the root cap, you can find region of meristematic activity, where here meristematic tissues are present and root apical meristem is present. Okay, root apical meristem is present, where the location of root apical meristem we will say it as subapical or subterminal. Proximal to the region of meristematic zone, you have region of elongation where cells will elongate and the length of root is elongated, and then the region of maturation, okay, maturation region where root hairs are formed, okay. In the maturation region you can observe root hairs. So what you have to remember is at apical or terminal you can find root cap, proximal to this what is their region of meristematic, proximal to this what is their elongation, proximal to this maturation zone. And then different types of the roots like tap roots, fibrous roots and adventitious roots. Prop roots, tap roots you can see in most of the dicots, fibrous roots in most of the monocots, adventitious roots you can observe in sugarcane, maize, prop roots like banyan, okay. So that you can clearly look into the root modifications once better. And this diagram of leaf where you can see the three parts of the leaf, those are petiole which is the stalk of the leaf, and midrib, and then lamina, leaf lamina, okay. These are the three parts of the leaf. At the region of the petiole, small bract-like structures which are there, those are called as stipules, and you can clearly observe reticulate venation which is seen in most of the dicots, parallel venation which is seen in most of the monocots. And here if you observe phyllotaxy, okay, types of the leaves, that is simple leaf and compound leaf. Compound leaf is of two types, that is pinnately compound and palmately compound. Pinnately compound where to the leaflets which is called as rachis, here it is called as rachis, to that leaflets are arranged in the successive order. Best example is neem. Palmately compound, at the tip of the apex, at the apex region you can find many leaves at the terminal point where you can observe. The best example is silk cotton. And then if you look into phyllotaxy, arrangement of leaves on the axis is called as phyllotaxy. Alternate pattern you can observe in China rose, mustard and sunflower where only one leaflet you can observe at each node, okay. Opposite if you see, at each node you can observe two leaflets. Best examples are Calotropis and Guava. World phyllotaxy, at from a single node you can observe more than two leaflets. Best examples include Nerium and Alstonia. Okay, you have to remember all the examples better, highly important. Okay, coming to racemose inflorescence and cymose inflorescence. Racemose inflorescence where the main axis is indeterminate and flowers are born in a acropetal manner, okay, acropetal succession, and flowers arise in centripetal manner, flowers arise in centripetal manner where you can observe young flowers at the top and older flowers at the bottom. If you look into cymose flower, you can see a basipetal order, okay, where the apex will terminate to a flower, okay. There is not indeterminate axis, it is a determinate axis where apex is terminating to a flower, and here you can clearly observe basipetal succession. Here you can clearly observe basipetal succession, and flowers arise centrifugally, okay. Flowers will arise centrifugally. Okay, that's it. And next if you see position of floral parts on the thalamus. Based on the position of the ovary with respect to the other floral parts, we have three types of flowers. Those are hypogynous, perigynous and epigynous flowers. Hypogynous flowers wherein ovary is superior and other floral parts are inferior, below the ovary position, where ovary is at the superior position, okay. Best examples include mustard, China rose and brinjal. Perigynous where ovary is half inferior and half superior. Last time this diagram is given and most of the students are confused. Need not to confuse at all, better you can see the other floral parts clearly at the rim of thalamus, right. Other floral parts are clearly present at the rim of the thalamus. Best examples include plum, rose and peach. Next, epigynous flower. Ovary is completely inferior where the thalamus is covering the ovary wall and other floral parts are above the ovary. Best examples include flowers of guava and cucumber, ray florets of sunflower. And then this is aestivation, highly important. Okay, this time we can expect a question from aestivation, either it is diagram based or example based. Okay, so yes, if the sepals and petals just touch each other, not overlapping but just touch each other, that is called as valvate aestivation. That is valvate aestivation. Second is, if one sepal or petal overlaps the other, then that is called as twisted aestivation. It is twisted aestivation. If they overlap but not in a particular direction, best example, that type is called as imbricate aestivation. That type is imbricate aestivation. And if you can observe one standard petal, two wing petals and two joint keel petals, that is called as vexillary aestivation. Okay. For valvate aestivation, best example is Calotropis. For vexillary aestivation, it is pea and bean. For imbricate, it is cassia and gulmour. And for twisted, it is cotton. Okay, do remember beta, aestivation is very important. And next, if you look into placentation, which is arrangement of ovules inside the ovary. Okay, if ovules are arranged along the ventral suture of the ovary, the best example is pea. And if ovules are arranged in the inner locule, forming chambers or locules inside the ovary, this is called as axile placentation, and examples include China rose, tomato and lemon. Parietal placentation, where ovules are formed at the inner walls of the ovary, examples include mustard and argemone. Free central placentation, where ovules are arranged in the central part of the ovary. Ovules are arranged in the central part of the ovary without forming any locules. Examples include dianthus and primrose. Basal placentation, where ovule is at the basal part. Best examples, this basal placentation is applicable to ovule, single ovule. Okay, single seeded fruits also you can say. Best examples include sunflower and marigold. And then if you see types of fruits, okay, wherein mango and coconut. This diagram is also very important. What is this type of fruit called as beta? Drupe, where pericarp has epicarp, mesocarp and endocarp. Here in the mango, which part is edible? Mesocarp is edible, but the same mesocarp in coconut is fibrous. Do remember, it is fibrous. And here you have stony endocarp, here also it is stony endocarp, here it is thin epicarp. Okay, this is the differentiation of the pericarp in mango and coconut. And then if you see dicot and monocot seed, dicot seed and monocot seed. Dicot seed, if you look into, you can clearly observe the embryonal axis, two cotyledons, and embryonal axis lower part differentiating to radical, higher upper part towards the apex, shoot apex, that is plumule. Okay, and the small scar like structure on the seed is hilum. If you look into monocot seed, which is very important, which is asked in the NCERT of 2025 also, so don't miss this at all, which is asked in the previous year NEET also. So yes, here this is endosperm, which is cellular endosperm, and this wall is aileron layer, which is proteinaceous endosperm. So here if you can see, this is cellular endosperm and this is proteinaceous, or you can also call it as aileron layer or proteinaceous endosperm, and this is papery embryo, that is called as scutellum. Okay, papery cotyledon in the monocotyledonous seed, that is scutellum. Embryonal axis towards the apex is called as plumule. Covering the plumule, the covering sheath above the plumule is coleoptile, and below part it is radical. The sheath which is covering the radical is coleorhiza. Coleoptile and coleorhiza you see only in the monocot seed. And in the previous year question also you got, wherein this region is radical which gives rise to the root, not coleorhiza, and this region at the shoot, that is plumule region, that gives rise to the shoot, okay, not coleoptile, to remember. Next, this is from anatomy of flowering plants, wherein just now we have discussed, this is root cap which is at the apical region. At subapical region you can see root apical meristem. Initials which are just above and below the root apical meristems, these are initials of central cylinder or cortex, and this is the central cylinder at the center. This gives rise to vascular bundles, and here next to the central cylinder you can see cortex which forms the ground tissue. Fine. Now coming to shoot apical region, here at the apex of the shoot you can clearly see shoot apex. Far from the shoot apex you can observe leaf primordia, you can observe leaf primordia, and here if it is the leaf primordia at the axil where the angle between leaf and shoot, okay, this axil will form axillary buds which is over here, okay, axillary buds, and these are differentiating vascular tissues. Clear? So shoot apical meristem is at the apex, here is the leaf primordia where the angle between the shoot apical meristem and leaf primordia, this is the region where axillary bud is formed, and this angle is called as axil, okay. Now simple permanent tissues, where we have to remember for sure, this is parenchymatous tissue where the wall layers are simple made up of cellulosic cell walls, and you can clearly observe the intercellular spaces between the cells. Here if you see collenchyma beta, you can observe the cell walls with the composition of cellulose, hemicellulose and pectin. This collenchyma is highly useful for petiole formation and it gives mechanical support to the plant. Next, coming to sclerenchyma, you have two types of sclerenchymatous tissue. One is fiber and second is sclerites. Sclerites you can observe in the fruit walls of nuts, and you can observe in the pulp of guava, okay, and you can also see it in the tea leaves, okay. Sclerites will have very narrow lumen when compared to fibers, okay. And then if you see tracheids and vessels, the long tubular structures, tube-like structures with tapering ends, tube-like structures with tapering ends and pits at the center, these are called as tracheids. Vessels are long tube-like structures with diameters, and here you can see perforation plates above and below, okay. These are the tracheophytic structures of xylem. When it comes to phloem, here at the center you can observe sieve tube cells, mature sieve tube cells, okay, mature sieve tube cells are enucleated, mature sieve tube cells are enucleated. Next to the sieve tube cells you can observe companion cells. Both sieve tube cells and companion cells are called as sister cells as they are ontogenetically related. Next to that you can clearly observe phloem fibers and phloem parenchyma. And then if you look into stomata, okay, here the stomata structure is also asked in the previous years, where these are the epidermis and this is the subsidiary cells, these are the guard cells, and at the center you can see stomatal pore. Stomatal pore, guard cells and subsidiary cells together form stomatal apparatus. They will form stomatal apparatus. Okay, and here you can see dumbbell shaped guard cells, means these are of monocotyledons. These are of monocots. Here you can see bean shaped guard cells, means these are of dicots. Okay, and then Here you can see the arrangement of vascular bundles. It is radial, where one xylem and phloem, they are not arranged in the same radius. This is called as radial arrangement. This is called as radial arrangement of xylem and phloem. Xylem and phloem. This you will see it in roots. This you will see it in roots. And here it is xylem and phloem which are at the same radius, where you will call it as conjoint, and as there is no cambium between xylem and phloem, it is closed. Okay, conjoint and closed. And then here if you see, Nana, xylem and phloem have a cambium in between. Okay, xylem and phloem have a cambium in between. This is conjoint again, conjoint and open form. Okay, conjoint and open type. And then here if you see, which is very important, that is dicot and monocot root. Okay, here it is dicot root and here it is monocot root. Just by looking into the diagram, how we can quickly say, because dicot root has large pith and monocot root has smaller pith in the stellar region, and here it is highly tetrarch, like you can see only 4 xylem and phloem strands. Here if you can see, there are more xylem and phloem. This is polyarch, xylem and phloem bundles in the stellar region. Clear? And then if you observe, Nana, this is dicot stem, wherein epidermis, next hypodermis is made up of collenchyma, and then it is parenchymatous general cortex. Then you can see clearly endodermis is filled with starch sheath, and semi-lunar shaped pericycle is observed, and phloem and xylem have a cambium in between, and at the lower part and at the center you can observe the pith. These are the semi-lunar patches of pericycle. Clear? Next, when it comes to monocot stem, here in the monocot stem you can observe epidermis followed by hypodermis. Hypodermis is made up of what cells? Sclerenchymatous hypodermis. It is sclerenchymatous hypodermis. Then completely it is filled with general cortex only. Sclerenchymatous hypodermis followed by general cortex, and vascular bundles which are present in this general cortex, these vascular bundles are of schizolysigenous origin. As vascular bundles are scattered in the cortical region, it is schizolysigenous in origin, we will call it as. And then quickly if we look into the leaf of dicot and monocot. In dicot leaf you can see adaxial epidermis and abaxial epidermis. Adaxial epidermis, if you see, Nana, at the above region, adaxial epidermis, here you can observe less stomata when compared to the abaxial epidermis. Adaxial epidermis, which is the ventral surface or the upper surface of dorsiventral leaf, abaxial surface, which is the lower surface of the dorsiventral leaf. Adaxial surface has less number of stomata when compared to the abaxial epidermis. Abaxial epidermis has more number of stomata. This adaptation is to escape the process of transpiration. And here you can see two types of mesophyll cells. One is palisade parenchyma, which is perpendicular to the adaxial epidermis. Next is spongy mesophyll tissue or spongy parenchyma. In between you can clearly observe the air cavities. Inside the spongy mesophyll cells you can see the vascular bundles also. You can see the vascular bundles which are again conjoint and closed. These vascular bundles are covered by bundle sheath cells whose cell walls are very thick and there are no intercellular spaces for bundle sheath cells which surround the vascular bundles. Next, coming to the monocot leaf, where you will call it as isobilateral leaf. Epidermis is same as that of dicot, but one structure is unique here, that is bulliform cells whose size of cells is larger than the epidermis. This bulliform cells will maintain the shape of the leaf both in turgid and flaccid conditions. They will maintain the shape of the leaf both in the turgid and flaccid conditions. And adjoining to the vascular bundles, you can see bundle sheath cells. Okay, these bundle sheath cells are up to the adaxial and abaxial surface of epidermis. Here you can clearly observe equal distribution of stomata in both upper and lower surface of the leaf. That is why it is isobilateral leaf. So here this type of arrangement of stomata is called as amphistomatic. It is called as what, Nana? Amphistomatic. And here this type is called as hypostomatic. This is hypostomatic. Okay, and here also you can see bundle sheath cells surrounding the vascular bundles. They go up to the adaxial and abaxial epidermis. So till this we have discussed today. In tomorrow's session we will discuss about all the diagrams in the second set of NCERT 11. So yes, stay tuned and thank you. All the very best for your exam.